So there's a second mechanism by which we transport oxygen, and that has evolved in organisms that are larger, and that is by bulk transport. What do I mean by bulk transport? Well, you pick up the medium, air or water, and you move it from one place to the next with the gas in that medium. This is often referred to as convection versus diffusion. It's bulk transport. So let's go through an example. We'll go through one that looks like a circulatory system, where the little blue dots represent a gas X. You could say it's oxygen, and that fluid is moving through the vessel at a particular flow rate, as an amount of that fluid per unit time. So we have our rate dot there. And there's a certain concentration that's in the red blood cells plus the surrounding fluid of that gas X, of let's say oxygen. And so how do you write the equation for gas transport for convection? Pretty straightforward. It's the amount of gas moving per unit time of gas X, which equals the flow rate, the amount of the fluid per unit time, times how much gas is in that fluid, the concentration of that fluid. Now, remember, we can write concentration of the fluid as the capacitance times the partial pressure. And when we do that, we can then take the two other terms added to partial pressure and call that conductance. In this case, it's conductance relative to convection, which is just the flow rate times the capacitance. Okay, but now you can see we have a general relationship that can be used for both diffusion and convection. And in fact, what we'll show in our model is that these are linked. And so it's very easy then to make the complete model by taking this simple transformation. And so how do we think about the flow rate part of the convection equation? Well, flow rate often equals the volume per pumped of the fluid per pump times the frequency of that. We also often think about the circulatory system when we do that, but you can also have it in the respiratory system, the breath you take per pump times the respiratory frequency. But so it can be written very general too. We can write it as the rate of flow of that medium, air or water, times the volume per pump times the frequency. And if we're talking about the circulatory system in sort of a more medically related kind of language, that would be called the cardiac output, the flow rate, times which equals the stroke volume, how much blood your heart pumps per beat, times your heart rate, your frequency. And so that gets the flow rate of your blood, and you can put that into your equation if you know the partial pressure changes the one end and the other from arterial to venous. You can get an estimate of that flow rate of your gas transport. Okay, so now those are the two sort of major systems. Let's put it in a general model now because of the way we've set it up, the way we've transformed it. And so on the left, I have my attempt at drawing an alveoli or a lung, and below we have a plot of partial pressure in torr that will follow through the whole system from the air all the way to the mitochondria. And we'll show how these simple relationships can be linked so that you can make predictions with respect to their overall design. So we start off with a convective step. Again, we can write it with our general model, and that's often, but not always, in organisms as the first stage of bringing in gas, and is referred to as ventilation. You can ventilate air like we do, or you can ventilate water like animals do over their gills by pumping their gills. But it's a bulk flow of fluid, air or water, with your gas in it to be delivered Now, if you look at our partial pressure of oxygen in the lung, then when it goes in and mixes, what you'll see is that the partial pressure begins to drop as it gets mixed with CO2, and you unload some of that oxygen into the deoxygenated blood coming back. What does that look like? Well, if you take a slice of a mammalian lung, there's the oxygen going through the bronchioles down to the little alveoli, and then you get to a point where what's the next step in our gas transport model? Well, you now have a barrier that you have to go through, and that would be a diffusive step. So we can take our simple equation again for diffusion and say, okay, you got to go from the alveoli into the circulatory system. So where G, conductance and ventilation, was the flow rate times the capacitance, now the G D for diffusion is going to be the area, the diffusion coefficient, times the capacitance divided by the thickness. It's just a different G, but still going to be limited by the partial pressure of your gas on one side of the membrane and the other. And so we represent it here by the circulatory system. So the deoxygenated blood, the light blue, is coming in, and the oxygenated blood, red, is going to go out, but we have that diffusive step. And then all the things we said were important about it, particularly trying to stop the limitation of that step by increasing surface area. So there's going to be a gradient of partial pressure as the deoxygenated blood passes through your respiratory organ. It's going to be low, and then it's going to get higher as it gets oxygenated. And so we can see that sort of the structure if we look at the wall of one of your alveoli or air sacs. So there's the where the oxygen is coming in through the alveoli into the diffusing into the capillary. Or if you take a section of it, you can see that this is a capillary with some red blood cells sitting into it. There's the membrane that it has to diffuse across, and the oxygen comes in and binds to a respiratory pigment, maybe hemoglobin or one of the other ones that animals have. Okay, so now we have that ability to load up our fluid, in this case blood, with some oxygen and begin to transport it again. So bulk transport is the next step. It's another convection step that's often referred to as perfusion, and that conductance then is similar to what we said for ventilatory conductance, and that is it's going to involve the flow rate of the fluid times its capacitance, how much is being held there, where the flow rate is going to usually depend on a pump, the volume per pump times its frequency to look at the delivery. And so you see what's the advantage of this is that we then can chain these together. And if these rates don't equal one another, then one step you'll see a depletion or an increase in the gas, right? So you can see the wonderful coordination that occurs in the system in order to deliver gas. And so then we can say for the PO2, the arterial blood is going to be higher if it gets oxygenated well, and the venous PO2 is going to be significantly lower as you dump off the oxygen into the tissues. All right, let's look a little bit more carefully at the oxygen or the gas in the blood that's going to be perfused by convection. And we're going to go back really to the curve we talked about when we showed all three, air, blood, and water. And we're going to come back to that curve that we showed blood with respiratory pigments. And we're going to talk about something referred to as the oxygen dissociation curve, which also could be called an oxygen association curve, because it works both ways. And often this is talked about in isolation, and you memorize it. We're putting it into the grand scheme of our oxygen transport model to really understand what the value is with respect to respiratory pigments. So we'll have a plot of PO2, like we did in that earlier diagram, as a function of concentration. This case, we'll talk about oxygen, the milliliters of oxygen per liter of blood. And then we'll say, well, what if you didn't have any respiratory pigments? How much dissolved oxygen would you get? And remember, we showed that curve in water in the original guy, and it's really low. It's really low. So if you only have dissolved oxygen, it's hard to imagine you could have a very high oxygen transport and high metabolic rates. What kind of difference do we see when we add respiratory pigments? It's an enormous difference when you include the oxygen-bound respiratory pigments like hemoglobin and hemocyanin. We'll talk about a few of them in a moment. But this then shows that as you increase the partial pressure, so it matters what the environmental partial pressure of oxygen is. When you get to the high level of PO2, you'll see that you get the greatest concentrations. And that's because you get to a point where all of the sites on the respiratory pigments are bound with oxygen. And that's about as much as you can carry then in the blood. And in fact, it's called carrying capacity. So one of the things you want to know about your blood is what's the carrying capacity, because that'll tell you the limitations about the amount of oxygen that you have available. So that's one really important parameter, carrying capacity. There's another one, but that one's really pretty critical. And it's going to depend on the partial pressure, but also your respiratory pigment, how much you have of it, and so forth. So carrying capacity. Now, we only think of hemoglobin, but in nature, there's lots of oxygen pigments. And in an odd way, they're beautiful, if you've ever seen any of them. And they're just extraordinary. So we see hemoglobin shown on the left there, the majority of vertebrates. But there's also blue blood. In fact, it's kind of cyan, light blue blood. And I've seen it in crustaceans when we've done experiments on their oxygen transport. And it's a beautiful light blue. I've also seen chlorocurarine in mostly in worms and some leeches, where they have green blood. And then there are other marine worms and some strange creatures that have purple blood, have hemierythrin. It just depends on the metal associated with it and the light reflection. But there's a bunch of different respiratory pigments that have different properties. All right, let's then use our comparative method and try to better understand our gas transport model by looking at the variation. And so let's compare the variation first in carrying capacity that we talked about. So we have a plot here, the same plot of oxygen concentration in millimolars per liter as a function of the partial pressure of oxygen for a bunch of different species. And what you can see immediately is that the warm-blooded mammal, humans, has very high carrying capacity compared to, for example, the crab that uses a different respiratory pigment, hemocyanin, that has a really low carrying capacity. And then other animals are sort of in the middle. And it's actually a function of temperature, pH, and even PCO2. We'll come back to that. But you can see that there's a lot of variation in what's possible with respect to aerobic metabolism and the activities that you can sustain on that with that approach, with that metabolic pathway. Okay, so in addition to carrying capacity, there's another key parameter that we need to understand in order to fully appreciate the oxygen dissociation curve and what the respiratory pigments can do for us. And the way we look at that is we say, okay, we're going to normalize the concentration, the carrying capacity, as 100%. If you've reached that carrying capacity, that's 100% saturation. And so we're going to compare percent saturations by normalizing all those different carrying capacities that I just showed you. And then we're going to make a measurement that's often made to then look more carefully at the shape of this sigmoid curve. And so we often say, okay, what's going on at 50% saturation. What's the partial pressure of oxygen at 50% saturation? That's referred to as the P50, and that's a measure of affinity, of how easily it grabs oxygen or how easily it dissociates and lets go of oxygen. And so our question is, how important is this? And what we can do is get a hint from, the comparative approach to diversity of organisms by making this normalization to percent saturation. Let's look at a bunch of these same creatures. We have now the percent saturation plotted against the partial pressure of oxygen for these same species. And then we can look at the P50. And what we see is that there's an incredible difference in the P50s. Now, how much sense does this make? Well, it's interesting, and it's dependent on, again, temperature, pH, and PCO2, so there's a lot involved in trying to standardize and normalize the comparison. But we can see pretty clear differences. For example, the lugworm, 2, has a very, very low P50. Therefore, its pigment has a very high affinity for oxygen. So if there's any oxygen out there in the hypoxic mud there, it's going to grab it. It's going to pull it in. Very tight affinity if any is out there, and then holds on to it. Whereas if we look at the far right, iguanas, they have a very high P50, and therefore they have a very low affinity. They have an advantage of being able, if they get enough oxygen in the air that they're in, to more easily unload that oxygen in the tissues if they can reach this kind of very high P50. So it kind of works both ways, and you have to understand both the environment as well as what's happening within the organism to interpret the affinities and whether they're advantageous or disadvantageous. So let's look at this a little bit more carefully. Let's take an example that you may be more familiar with. You go into the doctor, they stick a needle into you, they take some blood, and one of the things they might be measuring is your arterial PO2. And so if your percent saturation is normal, it's high 90s or 100%, then your PO2 may sit there where we have the arterial blood. Of course, if you then take a venous sample, that PO2 is going to be significantly lower because you've dumped off some of the oxygen to the tissues, and you'll see then the sigmoid curve, you move down the sigmoid curve, and it starts to drop significantly. And what we know then is that the difference between the arterial PO2 and the venous PO2 gives you an amount of oxygen that's been extracted by the tissues. And so these are two important measurements to understand what's going on with respect to oxygen delivery. Now, it's interesting in the design of the pigments that you might say, well, what conditions would favor a high affinity, that is, you really want to grab onto that oxygen, or would favor an unloading, a dumping off of that oxygen in the tissues. And so we can make predictions. We could say, okay, well, if you could do it and this pigment would be responsive to the conditions around it, what would you expect to be able to do to make it more advantageous at the lungs? Let's say if you get to a place where, because of the environment or disease, you have a lower PO2 available. What would you want to have happen? Well, just like the lugworm, you'd want to sort of decrease that P50 at the lung and load more oxygen. So you'd expect that to happen. You'd predict that would happen. Then your arterial PO2, instead of starting to drop to a lower percent saturation, would in fact be preserved. turns out if you look at the lung for hemoglobin like this, this is exactly what happens at the lung. You have a decreased P50, greater loading, so you have a shift to the left to load at the lung. One way to remember the direction of this. And then you say, well, what conditions favor this? What's going on at the lung? Well, we know the temperature is a little bit lower, the pH is a little bit higher, a little bit less acidic, and you have low CO2. You're getting rid of the CO2, dumping it off. It turns out that if you take your respiratory pigment in a flask and set these different conditions, you change the temperature, you lower the temperature, you increase the pH, you lower the, and you measure these curves as a function of partial pressure, you get that dashed line. This is happening with respect to the pigment itself, favoring loading of oxygen at the lung. And so now you can do the same thing to think about what happens at the tissue. So now you're loaded up with oxygen, you're now going to the tissues. What would you expect to be happening? What would be advantageous at the tissue? You have the opposite. You'd expect an increase in P50 to better unload that oxygen at the tissues. So in this case, your venous blood concentration of oxygen would be at the same PO2 would be much lower. Why is that advantageous? Because you extract more oxygen out of that blood if you had that shift. What conditions would favor that shift in the respiratory pigment? Well, it's the opposite. High temperature, low pH, more acidic, and a lot of CO2 that you're now getting rid of in the venous blood. And if you were then take your hemoglobin in your flask and test it under these conditions, this is exactly what you get. You get the P50 of the dashed line that's in this case shifted to the right. And so you can see that the affinity is another important parameter that can vary significantly and make a big difference with respect to how much oxygen becomes available for the diffusion pressure into the tissues. It can make a big difference. And so therefore you'd make other predictions too based on these principles, and you could test them. Here's one that is, I think, interesting, but from what we talked about is kind of predictable. Let's plot the percent saturation of your pigment, of hemoglobin in this case, against your oxygen partial pressure, and compare fetal hemoglobin oxygen dissociation curve with the maternal. Now, is this in the direction that you'd expect? It is, right? And it's because of some chemicals associated with the fetus, but the fetal hemoglobin has a higher affinity than the maternal, so that you can have a greater diffusion rate, greater PO2 across the membrane ultimately. So you're grabbing as much of the oxygen as possible in order to be able to have the fetus get that high diffusion rate from the maternal source. So that's kind of predictable. And then you can use the same kind of thinking even in a completely different circumstance. What about tadpole development into a frog? You can look at percent saturation of your pigment as a function of our oxygen partial pressure again, and say, what does the transformation look like as the tadpole grows up into a frog? And is this what you'd predict? Well, let's look at it. The tadpole affinity, when it's in the hypoxic ponds, is really high. So that's the left-hand curve, and also associated with particular pHs. And it has a very low P50, a high affinity. And then over time, as it becomes an adult frog, it shifts for greater unloading of lower affinity to be able to dump it to the tissues, and to correlate it again with these changes that we've talked about. So again, if you understand this sort of general model, you can make predictions ahead of time about a function, about what might be going on, and not only in these normal states, but it's really, again, powerful to predict disease states and all the things that might happen. You can also look at changes in body mass. What would be our prediction there? So here's percent saturation against oxygen partial pressure. Well, we talked about the fact that body size matters, right? It makes a big difference. And so here's a plot of the oxygen association curves under controlled conditions that compares animals of different size, from the elephant on the far left with a low P50 all the way to the little mouse. This is number 8, which has a high P50 or low affinity on the right. Now, does this match what we would imagine? Well, what we see is the affinity of mammalian hemoglobin decreases significantly with a decrease in body mass. So as you get smaller, it moves to the right. Why might that be? Well, we said if you take a piece of tissue of the same mass and compare an elephant with a mouse, the mouse tissue, same mass, has a much higher metabolic rate, right? Much higher metabolic rate. And so the small animals require greater oxygen extraction. And so you can imagine that's favored by a P50 that is increased where you have lower affinity releasing to the tissues. And that's what you see. Okay, so that helps us understand what's going on in perfusion in the convection system of moving blood, and the critical role of respiratory pigments for larger organisms and ones that are ultimately more active. But then we have another step, a final step, right? We have a diffusion step, and we have to then diffuse from the capillaries into the tissues all the way to the mitochondria. And you could say that's one big diffusion step. People often break it up into several smaller ones because they're different components of diffusion, but it's a diffusion nevertheless, and the things that we talked about for conductance to the tissues, G of T, make a difference, right? And so the area and the coefficient, the diffusion coefficient again, along with capacitance divided by the thickness, all those things still matter. They just may be better described and tailored by several diffusion steps to describe them more effectively. And so then you're going to get your PO2 that's going to drop almost to 0 by the time you get to the mitochondria. So you can see that in this kind of capillary network. Here's the arterial end, the blood's coming in, here are the capillaries. Now you have a diffusion step into the tissues, and then you have the venous end taking them away. And finally, you have a picture right down to the red blood cell. So the red blood cell in the hemoglobin has that oxygen, and it's releasing that to diffuse into the tissues. And therefore, you can imagine that geometry matters in terms of how it's set up. And we'll talk more about that later. But there's an example of the red blood cell in the slice, and the mitochondria in the tissue. So you can see how they're associated with the red blood cell in close association. So again, that diffusive step is reduced with respect to thickness or with respect to length. That you can put the mitochondria in different positions to facilitate that final diffusive step. Okay, so that's the first part of our gas transport model. Next time we'll talk about it more generally with some other general principles, and we'll give a bunch of examples. We'll run through the diversity of organisms with their gas transport systems.